All right, let's get started. Well, thank you all for joining today's didactic training session. Just a quick reminder, we're recording the session and we will post it to the Transcend website for future viewing. And then after the presentation today, we'll have time for question and answers. You can either respond or ask questions directly or add the questions in the chat. And then at the end of today's presentation, we'll also share a link to a short survey. Your feedback is extremely helpful in shaping future professional development, so we do appreciate you taking a couple minutes to complete the survey. And with that, we'll get started. Welcome to today's Transcend Professional Development Core didactic training titled Power Analysis Made Manageable. We're fortunate to have Dr. Mark Williamson leading today's presentation. Dr. Williamson is an assistant research professor at the University of North Dakota School of Medicine and Health Sciences for the Population Health Department. He serves as a statistician and coordinator for TRANSCEND's Research Design, Compliance, and Data Management Core. He has a broad scientific background that includes physics, analytical chemistry, biomedical research, statistics, ecology, genomics, and epidemiology. His current research is exploring radon exposure as a risk factor for various cancers and developing experimental radon projects, which include small radon chambers for remediation testing, cell culture exposures to radon, and an experimental radon test chamber for animal experiments. Please welcome Dr. Williamson.
All right. Hello, everyone. Thanks so much for joining this didactic lecture series on power analysis. So let's get started. So the objectives of this training session is to understand the importance of power analysis, to describe the information needed for power analysis. What do you need to actually bring together to run one? And then I'll walk through power analysis for several common statistical designs to kind of show you how it works. And maybe most importantly, help develop a strategy for more complicated statistical designs if your study goes above and beyond maybe a simpler analyses. So what I really hope, is to have power analysis without too many tiers throughout this. So it's good to start maybe with what is power analysis, a bit of a definition. A good functional definition that works for what most people actually want to get out of it, I would describe as being a method to determine the appropriate number of samples for a proposed study. So it's getting sample size, whether that's rats, whether that's human patients, whether that's cell cultures, whether that's ecological plots, whatever. Now, if you wanted that sort of more complete, exhaustive, nerdy, me pushing up my glasses and saying, well, actually, it'd be something along the lines of statistical method to determine sample size, effect size, power, yada, yada, yada. We're trying to make this manageable, so let's just stick with the functional definition. We could go to more details, but really what most people want out of power analysis is sample size. Now, it's not the only thing you can do to sort of get an estimation for sample size. There's something called the resource use equation that's sometimes used for animal studies when you don't really have a lot of pilot information. And you can also sort of supplement power analysis by things like literature comparison. Look at similar studies. How many samples did they use? Did that seem to be enough? That might not be the best on its own, but it's certainly great to sort of ground maybe some of your calculations in what people have actually found and found success with in real life. And you can also do more complicated things like analyzing with a simulated data set. Again, probably not its own, but you can do that with the power analysis. So why? Why do power analysis? Why get sample size? At the broadest level, it's the idea of good stewardship. It's the idea of wisdom. It's the idea of prudence in your activities. And, that's a broad, old idea. We see in the Gospel of Luke, we see this passage, Luke 14. Which of you wishing to construct a tower does not first sit down and calculate the cost to see if there's enough for its completion? Otherwise, after laying the foundation, finding himself unable to finish the work, the onlookers should laugh at him and say, this one began to build but did not have the resources to finish. Now, if you wanted to tweak that slightly for the more power analysis side of things, you could adapt that and say something along the lines of, which of you wishing to conduct an experiment does not first sit down and calculate the sample size to see if there's enough for completion? So the core of it, sample size, power analysis, it's about good stewardship, it's about wisdom, it's about prudence. Now, there's more specific scientific and ethical rationales for it. A big one on the scientific side is if you don't have enough samples, you have too few samples, you're likely not to really detect your expected effect. Or even if you do, you might have an underpowered study. And so if you don't have enough samples, you're kind of wasting your time. On the ethical rationale, you also don't want to have too many samples because that again is a waste of resources. You're subjecting more animal models to treatments and sacrificing, and you could put more human subjects at risk than necessary. If it's experimental treatment, maybe there's side effects. If you can get by with a smaller sample size to really see if it works or not, that's better than just testing a whole bunch of people with unknown side effects or more serious side effects than they need. And you could also make the ethical case that it's not good to have too few. Same with the scientific rationale. If you can't actually answer your scientific question, you're wasting your resources, you're subjecting patients unnecessarily, and that's not really ethically good either. And you could even turn more broadly speaking to science as a whole. So the idea of underpowered studies would be studies without enough samples to really detect a significant effect that should be there. There's a famous essay called "Why Most Published Research Findings Are False" by Dr. Ioannidis. Goes into maybe like 1 in every 2 biomedical studies that show a significant effect. That effect probably really isn't there. And there's a myriad of issues with that. But one of the things that pops out is that the probability that a research claim is true might depend on study power as one of them. So if you have an underpowered study, you might be contributing to the fact that there's a reproducibility crisis in a lot of areas of science. So with a quasi biblical homily and an exhortation that science itself depends on you for power analysis, hopefully I've helped set the stage for maybe why you should consider getting appropriate number of samples for your study. All right, now let's turn to the quantities needed. Like, what do you actually need to conduct a power analysis? So really, there's 4 things that go into power analysis. For most people, kind of giving our functional definition, we're looking to get sample size out, the number of observations per group. And for that, you need 3 other main quantities. You need your effect size. That's going to be the magnitude of the difference or relationship you're trying to detect. So if you imagine you have 2 groups, you have a treatment group and you have a control group, and you're looking at that means, maybe it's tumor size, maybe it's some sort of biomarker. You're looking at the difference between those 2 groups. The bigger the difference, the bigger the effect size, and the easier it is to actually detect that, and the smaller number of samples you need to detect that. So effect size is really big on this. Another 2 variables that you need to have is significance level, also known as alpha. If you're familiar with statistical tests, usually you have a p-value that is significant or not, depending on whether reaches this alpha. And different fields will have different settings. The most common setting for an alpha is 0.05. And so a lot of times with these studies, that's just the default, and that's what I usually do. So it usually doesn't take too much more than just setting it as the default, unless there's other circumstances. Then there's power. In fact, that's where we get power from power analysis. And it's the probability of correctly rejecting a false null hypothesis. You actually want to make this as large as you can, though, of course, the higher the power, the more samples you need. And so typically a good threshold that the default is usually 0.8. That's a good balance between the number of samples you actually need and making sure your study is well powered. Now, again, different studies, different types of studies might have different thresholds. Like in this example here, 0.95 is maybe more typical for clinical trials. You want to have more assurity. Of course, that will require more samples. But essentially, if you have these three things, plus maybe a few other details, effect size, significance level, and power, you can calculate sample size. And indeed, if you have any one of these three, you can calculate the fourth. So that's why maybe a power analysis of a different kind might be to actually calculate power. You could say, I have a known sample size, I have a known significance level, I have a known effect size, I can calculate power. And vice versa. You could calculate effect size or significance level. So that's the big quantities that go into a power analysis. So what do you do with those quantities? What are the steps to go into this? Well, the first major step is to actually figure out what statistical test you will be using. Are you going to, with your data, are you going to do a t-test, you're going to do ANOVA, regression, etc.? Because essentially every statistical test will have a different sort of power analysis to go along with it. There's not really a one size fit all. So you have to tailor that to whatever test you need to do in the end. Once you figure that out, you're going to need to estimate the effect size for that particular power analysis. You also want to gather your other inputs, your alpha, your power, etc. And then you're going to most likely plug those inputs into some sort of power analysis software. There are some things you can, and for simpler tests, you can calculate some of these things by hand, but often you're going to be plugging those in and having the computer run it for you. And then you're going to interpret whatever those results are. So this might look like, for a basic example, maybe you want to do a two-sample t-test. Again, maybe you have two groups of mice, a control group, a treatment group. You're looking at a difference between maybe tumor size, and you need an effect size. For a t-test, the effect size measure is called Cohen's d. It's sometimes abbreviated to be D. And let's just say for our sample here, we think it's going to be a minimum effect size, so it's going to be 0.5. We're going to set power and alpha to be the defaults, 0.8 and 0.05, and then something called the ratio. Essentially, we're going to have the same number of rats in the control group as the treatment group. So the ratio is going to be 1:1 or 1. We're going to plug those inputs into something like G-Power, a free software for power analysis. With all that information, it's going to spit out that I will need 126 total, or 64 per group. So that's what power analysis will look like. And because each statistical test will have a different calculation, will have a different effect size, a different measure effect size, and that's usually, the effect size is based on usually information about the data you're going to be collecting. That is usually the most work when it comes to setting up a power analysis. And so let's turn our attention to a little bit more about how you actually estimate effect size. So what are the ins and outs of effect size? Well, more broadly speaking, when we're thinking about writing statistical tests, whether it's a t-test, ANOVA, etc., you're generating with your test a test statistic, which is going to be a signal over noise ratio. And the larger your test statistic, the more likely it is to be significant. The bigger the effect size. And so really that's what effect size is. It's also a signal to noise ratio. It's usually not exactly the same as a test statistic, but it's usually related. So if you're running a t-test and you have a large t-value, t-statistic, you're going to have a large effect size. And so, again, as I mentioned, every statistical test is going to have a different effect size measure, and there's different equations for each effect size. It will take different variables. And again, because we're trying to make this manageable, thankfully, a lot of this you don't have to figure out yourself, because if you get the basic information, the computer will do it for you. So I'm going to kind of just say, thanks, computers, and abstract all that more complicated, scary equations that a lot of times I don't even know how to do myself. But let's look at maybe one of the simpler examples that we can get our head around a little bit. So it's for a t-test. It's pretty straightforward. That's a comparison between the means of two groups. When you run a t-test, you generate a t-value. When you're doing effect size calculations, the measurement, as we've kind of seen before, is Cohen's d, also abbreviated as d. And actually, the effect size equation is simple enough that you could often run by hand. Really, what you need is two means. So I just have the Greek letters mu here. That's often used as abbreviation for mean. So you need the difference between two means above the pooled standard deviation. So this is again a signal to noise ratio. Your signal is going to be the difference between your means, say between a control and a treatment group, how big that is, and then the noise is going to be how much variation is in the data, often captured by standard deviation. And so that's how you would get an effect size for something like a Cohen's d. And so again, that's pretty abstract. I'm using equations. Let's try to help visualize that. So I simulate some data here, a difference between two groups. Let's just say this is our control and our treatment. The red is the treatment, the green is control. And essentially, I can set the means between these two. The control, the green is going to be a mean of 1. I can set the standard deviations. And then the only thing I'm going to be varying is the mean of this treatment group, this red. And that's going to keep going up and up. And because Cohen's d is the difference between means and then divided by standard deviation, I can calculate the effect size for this hypothetical data. And this is actually a tiny effect size. It would take a lot of samples to detect whether this difference is real or not. Now, if I bump up the mean of the treatment group, the red, to such a level that the effect size is 0.20, that's a small effect. You can start to see it separate a little bit more. If we go to medium, we're starting to see the mean separate even more. Now, it would take a lot less samples than the tiny effect size to detect a difference. So the effect size is getting bigger. It's getting easier to detect smaller samples. This is a large effect. You really start to see those separating out. Even by eye, it kind of seems like, yeah, it looks like that experimental group has a higher mean. And if you go to something huge, like where the mean is 2 versus 1, and I think the standard deviation is 0.5, we get, a pretty big difference. You wouldn't need a lot of samples to really detect an effect this large. So hopefully in visualizing that kind of helps us see the differences in effect size and how that'll affect how many samples we actually need to get. All right, so let's talk about determining effect size. So we'll need to get certain information from our potential data, because obviously we haven't run the study yet. Otherwise, we'd run a statistical test. But we can get information from other sources on like what we expect our data to look like. So I kind of have a bit of a flow chart of how to find that information. So first question you might ask is, do you have pilot data? Did you run a small pilot study and now you want to do a bigger study? If yes, that would kind of be the gold standard. Then you can calculate effect size from data. For a t-test, you just need the means of your two groups and the standard deviation. You'd be good to go. Now, if you don't have pilot data, you could ask, well, is there a similar study in the literature, or maybe a similar study that you've done that's not exactly the same, but comparable? And if yes, you could get information, get means, get standard deviations from that data, and calculate effect size. Now, maybe it's no. You could still ask, well, are there relevant information on potential study results? Maybe you have some expertise in the area, so generally speaking, what kind of effect that should come here. Like maybe you're doing treatments with cancer and you know that a good cancer drug should, cut down a tumor size by half. And so you could use that sort of information. And you could couple that together and calculate effect size from that info. And then, all else fails, you have no idea, you're just kind of shooting in the dark. You could use something like an effect size table and get a very rough estimate of effect size, something like small effect size, medium effect size, or large effect size, and go from there. So those are our 4 ways. Calculate from pilot data, from the literature or similar studies, from just general expertise, or more or less an estimated guess without any prior information. So if you get information from there, you can plug it in and get effect size, get sample size. But now it's quick, take a pause here and realize the elephant in the room. We're going to need to plug this in somewhere. We're not going to do it all by hand. So the kind of the elephant in the room here is, what software do we need before we get going on that? So let's have a brief interlude to talk about that software. So there's a variety of tools you can use to calculate power, to calculate sample size, given our information. Now there's G-Power, that's a free software that I talked about before. There's R that has packages that you can install that have varying levels of power analysis, or you could have a paid software like Pass. There's pros and cons to all of that, but especially for maybe this group who are maybe interested in sort of dipping their toes in a little bit more, G-Power might be a good example because it's free. It's not always the most user friendly, but there's a manual. There is some tools, so I'll be using G-Power for the rest of this talk, but know that there are more options out there. So a little bit on G-Power. You can download it. It'll have a user interface with a variety of different options. Sometimes that's the hardest thing to it is figuring out which option because again, just get in your head, each statistical test requires a different power analysis, require different information for effect size. And so you need to figure out what test family, and then within each test family, which specific statistical test. You need to figure out the type of power analysis. Again, I kind of abstracted, or not abstracted away, but kind of simplified. Turns out there's 4 different types of power analysis. It depends on which one you want to get out. The functional understanding, the functional description was we want sample size. So G-Power calls that a priori, where we calculate required sample size given those other 3 metrics. So we can typically just keep that set like there. The big thing is we're going to need input parameters. We see alpha, we see power, other things. Effect size is going to be the big one. The nice thing is usually we'll have a button called determine. We can actually determine effect size. We can plug in things like means and standard deviations and it'll calculate it for you. Or you could just enter things directly if you already know the effect size. And then, of course, what you actually want out of it is going to be the output parameters. There's a lot of parameters, but typically what we're interested in is, well, what's the sample size? So total or group sample size. So kind of zooming in here, again, each power analysis will depend on the statistical test. In G-Power, there's 5 different test families and then a myriad of different statistical tests. Later on in the session here, I'll be going over, I think, 7 of the most common tests that you might work with: correlation, proportions, ANOVA, repeated measures ANOVA or RM ANOVA, a 2-sample t-test, regression, and chi-square goodness of fit test. All right, so we covered a little bit about G-Power. The software will actually be plugging our results from effect size in. Now, I think we can get back to the effect size. We've covered that enough. So 4 different ways of calculating effect size. Again, it's essentially where you're drawing that information to calculate effect size. So I'm going to do some quick examples based on some of my data on what this might look like. So if I have sort of the gold standard, I have pilot data. So I do some work on radon, where I have boxes generating radon, and I look at treatments to see if there's a difference in radon. For all my simple tests, I had a treatment which I had activated charcoal, which actually absorbs radon. It makes it go down. And then I had a blank, so I did nothing. And I have the differences in radon before and after my treatment. So if it's negative, that means the radon went down. If it's positive, it meant it went up. And so I have this raw data. It's very small pilot site, just 4 samples each. But then I can get my means and my pooled standard deviation to run a power analysis for a 2-sample t-test, which again, 2-sample t-test, you have 2 groups, like a control and a treatment, and you're looking at difference in means. And thankfully, with effect size for a t-test is fairly simple. Cohen's d is just the difference in means and pooled standard deviation. So you can actually calculate that by hand, which I'll do here just kind of to show how it works. So I take my 2 means, 1.97 and negative 0.65 in the top, divide by my pooled standard deviation. Now do that, and I end up with a Cohen's d of 1.73, which is actually a really large effect size, which isn't surprising, where I'm looking at difference in means. One mean is almost 2, and the other one is negative 0.65. So that's a really big difference. It should not take a lot of samples to detect this effect. So say I didn't actually have the pilot study, but I had a similar study or a study in the literature where I did the same sort of thing with more treatments, but I do still have this gray bar that's activated charcoal, and then the white is the none, the blank. And maybe I thoughtfully wrote that up in a way that a statistician could pull out the information and my means and standard deviation so I can calculate effect size. So again, difference in means, 0.93 and negative 2.29. I have to do a little bit more with the standard deviation because I essentially need to pool those together and divide by 2, which will give me a, a Cohen's d of 5.29, which is a huge, huge, huge effect size. Not surprising given you see here the non-treatment, the radon went up, whereas the charcoal, it went way down. So it's really easy to tell that those two groups are different. Will not take a lot of samples to detect that. So, but say I didn't have any of that, but I have some sort of extra expertise in the state of putting radon and testing things in boxes. And maybe I say to myself, well, I expect this kind of test to have a threefold effect with a standard deviation of maybe half of the lowest mean. And so sort of calculating or transferring that word problem into actual math, most people's least favorite thing in math. We know that we need this difference in mean. So a threefold effect might be, one mean is 3, the other mean is 1. And then a standard deviation, half of the lowest mean. So, well, 1/2 of 1 is 1/2. I can calculate up and I get a Cohen's d of 1.0. Also a large effect, though not as large as those actual pilot data or similar data. And again, maybe the least desirable, but maybe that's the position you're in. You have no information. You don't know how much the effect size is going to be. You have no pilot information to draw on. So you could use something like an effect size table. Again, different tests will have different effect sizes, but for a simplified table here, we know I'm running a t-test for Cohen's d, and maybe I think there's going to be a large effect size, so I could just select my d to be 0.8. So with that, I had 4 different sort of strata, 4 different ways to determine effect size based on what data I had. I was calculating the same thing, difference in means divided by standard deviation, but how I got that information changed. So for the first one, I had pilot data. The D was 1.73. The second was from a similar study, 5.29. I used my expertise for the third one to get a D of point R1. And then I had to guess because I had no information hypothetically for the fourth one, and I get a D of 0.8. So now let's plug that into G power to kind of see how that translates into power or into sample size. So if I have my G power here, again, we're just setting alpha and power to their defaults, also that allocation ratio. So I'm going to have the same number of samples in my treatment group and my control group. So that's going to be 1. And if I plug in my effect size D, 1.73, it's going to give me a total sample size of 14 or 7 per group. So very big effect size, did not take a lot of samples at all to detect a real difference. Now, for this next one, I have even bigger effect size. So unsurprisingly, it takes even less. It takes only 6 total or 3 samples per group to detect such a huge effect. Now going to my third one here, my D was 1.0. It's still a large effect, but much smaller than the other ones. So what I find here, again, plugging effect size D of 1.0, the total sample size is 34 or 17 per group here. And then if I had no information other than sort of an educated guess, I would get a sample size of 52 or 26 per group. So you can see what the effect size estimated is going to make a huge difference on the number of samples. In my case here, is actually helpful to use that pilot data because it turns out there looked to be a huge effect size. So using that information would be better for another study than just guessing because I would need a lot less samples. But sometimes you're not always in that space. All right, so that's how to get effect size. So I've talked a little bit about some of the other settings for some of these tests. You'll need to maybe deal with some of the other input parameters. I'll just talk about t-tests and ANOVA because that's often where you get the most um, tests you're running, and you need to think about this. So in a t-test, one of the other input parameters is tailed, so whether one or two tailed. And I typically set this as a two-tailed test unless you have a strong justification for a one-tailed test. A two-tailed test will take more samples, but it tests in both directions. So it's a little bit more justifiable. And again, I've brought this up a couple times, but the allocation ratio is going to be the ratio of your samples to your control and your treatment, or your two different groups. Typically, this is you probably have equal group number, and so you just need to put your ratio at one. But if you had another design where, say, maybe you had twice as many controls as your treatments, this ratio would change. For ANOVA, there's a couple additional things that you might need to plug in here. For just any sort of ANOVA, you're going to be the number of groups. So that's the number of categorical predictor variables. So in a t-test, there's two groups, treatment, control, whatever. In ANOVA, it could be, treatment one, treatment two, control, maybe positive control, negative control. You could have multiple different groups. And then if you're looking at like a repeated measures ANOVA, which is often common in biomedical research, you also need the number of what's called the number of measurements. So that's what's your number of repeated measures? Maybe you're doing 4 repeated measures, you're doing measurements at baseline, then you're measuring again at 1 month, 2 month, 3 month. So you need to put that information in. And then another thing is going to be for repeated measures is something called correlation among repeated measures. So again, if you take like a blood draw from a patient at baseline and another blood draw at 1 month, that patient, because it's the same patient, those blood draws are going to be correlated. And the strength of the correlation is going to matter for sample size. Now, if you have some estimate of that, how correlated it is, great. Otherwise, you could probably set it for a default of 0.5, at least you have better information. And I guess that brings up an important point here is a lot of times you're making your best guess or estimation of some of these parameters, and you might not know very well. And so it might not be the best approach to just run a single power analysis, with a single set of parameters. You might run a variety of tests where you have actually a range of maybe correlation or a range of effect size to see a range of sample sizes that might be realistically for your study. So keep that in mind. Throughout this training and for the later examples, I'm using sort of point estimates where I don't have a range, but know that that's totally something you can do and often is a good approach given real world uncertainty in design problems. All right, so with that, we kind of went through the why, the how, and now let's talk through a variety of some common examples. So 2-sample t-test, one-way ANOVA, repeated measures ANOVA, correlation, simple linear regression, chi-square test, and proportions. Now, for either of these, I'll have a research question and some probably fabricated data just to show a point. I could have done very boring, maybe real-world examples, but for the sake of myself and maybe all of us, I tried to have fun with it. So just bear with me on some of these examples. So we'll start with a 2-sample t-test, and my question is going to be differences in North Dakota and South Dakota school mascots. So the question I asked is, are high school mascots cooler in North Dakota versus South Dakota? So I took a random sample of 5 schools per state. Here's North Dakota, here's South Dakota. Got the information. In North Dakota, we have the Nighthawks, the Spartans, the Rockets, the Rough Riders, and the Eagles. South Dakota, we have the Pirates, Bulldogs, Golden Eagles, Moguls, and the Arrows. And then I gave each a score by how cool I thought it is. So it could be positive. So like the best scoring one in North Dakota of the sample, I thought the Rough Riders were the coolest. It could be negative. So I thought the Eagles were a little boring. Same on the side here. And so then that's kind of my mock data here. And with this data, I can run a power analysis by getting means and standard deviation. So let's do that. Again, here's my data here. I can get my means of coolness score between North Dakota and South Dakota. You see that it's actually very close. And there's a lot of variability, large pooled standard deviation. So because Cohen's d is pretty easy to calculate, we've seen that before. I can just do that by hand here just to kind of show it. It gives me a d of 0.16, which is a very small effect. So if I plug that into G power, and now I'm going to kind of show using the determine button. You can see here this, if you click determine, it'll actually have a little pop out. And this is typically where you can enter things like means and standard deviations if you don't want to calculate yourself. So I put my two means in and my standard deviation. I'll click calculate and it'll give me my effect size, which turned to be 0.16. So same as how I calculate by hand. So maybe I'm not crazy. I can calculate, it'll transfer to main window, and then given all my other sort of defaults, I'll click calculate and it'll give me a total sample size of 1,168 or 584 per state, which is very funny because there are certainly not 584 high schools in each state. So if I really wanted to test the difference between this, I would actually leave the realm of statistics and just sample all the schools and get my coolness factor and just know the difference for sure. All right, so that's a two sample t-test. Let's switch over to one-way ANOVA, where I'm looking at more than two groups. And my question here is going to be, do different types of energy drinks increase the rate of youth bike ramp jumps? So maybe, kid has an energy drink and just has, that increases their desire to just, take their bike off any old ramp they can find here. And so I've created a mock graph where Y variable is the bike ramping rate and then the drink type. So sort of our negative control is something bland like milk. Then maybe we have a sugar only treatment, a sugar and caffeine treatment, and then like a sugar, caffeine, and branding treatment, with the Eagle, the full throttle, much more bike rampy than even just sort of the sugar and caffeine, but otherwise sort of branded agnostic type of drink. So with this, for ANOVA, we're going to need, again, means and some measure of standard deviation. So I pull my means out from this graph here, pretending like this was real data. And then I just put standard deviation as 2, again, mocking this up. But if you had real data, you'd pull that out. Again, I can go to my G power. Now I'm going to go under F test for ANOVA, fixed effects, omnibus one way. Some of this gets a little confusing, but this is just a one-way ANOVA. I can determine. Now I'll have 4 different groups, put my means in, my standard deviation, get an effect size F, calculate over here. It'll give me a total sample size of 12 or 3 per group. So again, not surprising given the huge difference in the bike ramping rate between milk and the treatment with all the different things here, sugar, caffeine, and brandy. All right. What if I want to do our repeated measures ANOVA? So that's ANOVA, but you're measuring more than once. You're measuring over time or some other condition. So say I'm looking at blood levels of a fictional protein called RR39 and wonder if they differ across age groups after a repeated exposure to Rick Astley's song, Never Gonna Give You Up. And so, again, another mock graph here. I'm measuring this protein level in the blood of RR39, codenamed Rickroll. And we can see here across my different age groups, the oldest age group looks like increased protein levels at each exposure. For the lower age group, it sort of remains flat. And for the middle age group, it kind of goes up a little bit before decreasing sharply. But is this difference between groups significant? So I can test that. Repeated measures ANOVA. Again, I'm going to need means of those groups, a standard deviation. Under the F test, this is going to be a ANOVA repeated measures between factors. There's different things you can look at for repeated measures, whether it's across factors, between factors. Between factors essentially means is there a difference between groups? And so that's what I want to do here. I'm going to need to have my standard deviation within group, like the other ANOVA, my number of measurements. So 4, we have 4 time points, my group means, and then I'm going to just default my correlation among repeated measures as 0.5, given over information. That's going to calculate an effect size. When I click this, it's going to give me a total sample size of 294, which if you divide that by 12, so 3 groups times 4 treatment times, that gives us 24.5, but you can't have half a person, so I'm going to round that up to, I need 25 members per group and then per time. All right, maybe let's switch over to correlation, 2 numerical variables. Maybe I can ask the question, are the number of Nicolas Cage movies and the number of pool drownings by year correlated? So this comes from sort of a semi-famous in statistical tests or training about how correlation does not imply causation. So there's a website called Spurious Correlations would take 2 variables across time that clearly aren't like causal. Like it's clear that the number of Nicolas Cage, our films Nicolas Cage appear in, does not cause pool drownings. But they are correlated. If you look across time, you can actually get a correlation coefficient of 0.666. And so for a effect size for power analysis, for something like correlation, you need correlation coefficient, also known as R, or you could have an R squared, which is just that squared. In G-Power, it's going to be one way to do it is under the exact test family. You can do correlation. And you're going to need to compare this against sort of one correlation versus another. So say I default my null correlation is 0.5, and I want to see if something like 0.67 is significantly, like how many samples to significantly detect that difference between the two. And so I could either, in the determine, I could either have my coefficient of determination, so the R squared, 0.45, and it'll calculate essentially the R, the coefficient of correlation, or correlation coefficient, or I could just plug that in directly. In either case, it'll give me a total sample size of 116. So again, since this is against year, I need 116 years of data. In this case, I'm probably not going to be able to get that much information unless Nicolas Cage is semi-immortal. All right, let's switch to regression, which does try to look at potential causation. And typically, yeah, you have a Y variable and you have one or more X variables that you think might predict that Y variable. So maybe I want to take a bunch of sort of health phrases and test them a little more rigorously. We've all heard that an apple a day keeps the doctor away, or laughter is the best medicine, or even kill them with kindness. But what if we took those seriously? Could a measure of health, like length of hospital stay after an operation, could that be predicted by laughter level, apple number, and kindness exposure? We could create a model modeling that. So length of stay as a function of laughter, apple status, and then kindness. I could run a regression model. Essentially, what we see here is that none of this is significant, but laughter and apple is negatively associated. That means, the more laughter or the more apple you have, the shorter your hospital stay, you get better faster. And then kindness is positively associated. So kill them with kindness. The more kindness you're exposed to, the longer you have to stay in the hospital, the worse your morbidity is. Now, at least for G-Power, what I need is going to be an R squared. So I can get an overall model adjusted R squared. That's going to be 0.54 for this. But actually what G-Power needs is what's called a partial R squared. Not going to go into too much detail, but it's looking at the how much like each individual variable kind of contributes to the model. And so I can run some R code that I didn't show here to get partial R squared. It turns out the one that's the largest is going to be apple. So let's just take that as our variable to maybe look at. That's the strongest. So we're going to go t-test, linear multiple regression, fixed model, a single regression coefficient. So we're going to look at, apple, and then, based on having other variables in there. So since we have three predictor variables, laughter, apple, and kindness, we'll put the number of predictors there. But then under determine, I need a partial R squared. So I'll plug in my largest one for apple, 0.21. It's going to give me an effect size. It'll go here. It tells me I need a total sample size of 32. So this would be 32 patients to test this effect. All right. Well, last two here are going to be going to count data, where we're looking at things with like proportions. So there's a chi-square goodness of fit test. I'm going to ask the question. So my kids are getting a little older. They're getting into Pokemon cards, and there's different rarity of Pokemon cards. And I might ask, is there the observed ratio of Pokemon card rarities match the expected ratio in a 10 card pack? So they come in 10 card packs. There's common cards, uncommon, rare cards, and say maybe the pack claims that you should have expected ratios of 5 common, so half of them should be common, to 3 uncommon, to 2 rare, and the rares are probably the ones you want more. But then say I open 10 card packs, I get these observed counts, I can turn those into ratios, and I see here that my observed ratio of rare cards, the more desirable ones, is only 1.2. That seems a lot lower than 2. How many samples do I need to actually take to actually test for sure whether or not my ratios are significantly different from the expected ones? Well, with that, we're going to need to get out proportions from our ratios. And so we have our expected ratios and observed ratios. So our expected ratios is going to be our null hypothesis. And so I can take the ratios, divide them by 10, because there's 10 total here. So the first one for a common is 0.50, so half of them, 0.3, 0.2, and then my observed, I calculate the ratios here, or proportions here. This is going to be a chi-square test, good as a fit test. Can determine, so I have 3 cells, because I have 3 different categories. I have my null and my alternative proportions. I'll calculate an effect size of 0.20 and some change, and it tells me I have a total sample size of 295. Now, given this setting here, packs only come in packs of 10, so I would actually need 300 total to test this, so I need 30 packs. I'd have to round up. And if I had 30 packs, I could run a test to see if my observed ratios are significantly different from my expected ratios or not. And finally, proportions, going to be very similar, though usually a little simpler. So I'm going to ask the questions. Dolphin attacks, they're rare, but they do happen against humans. I want to ask, is the proportion of dolphin attacks on humans higher for females than males? Maybe dolphins are not only, aggressive, maybe they're sexist. Who knows? And so I'm going to get some mock proportion data. Say I've got observations of 20 attacks on humans. I find 14 have been against females and 6 against males. Now, what I'll need for a proportion test in G-Power is I'm going to need 2 different proportions. So now they call that proportion 1. That's going to be my null hypothesis. So if there's no real difference in males versus females, I'd expect a proportion of 0.5, so 50-50 chance. And then I can calculate proportion 2, the one I'm testing against, with my data here. So 14 females out of 20 is going to be 0.70, so 70%. So I want to test if that would be significant. So I can go to exact proportion difference from constant, and then I can plug in my 2 different proportions. It'll give me effect size, and it'll calculate out that I need a total of 49 samples, or 49 observations in this case, to test that proportion against the null. All right, with that, we've gone through some examples. I'll kind of close out here with some next steps. So hopefully this kind of gave you an introduction, maybe even give you some tools to do it yourself. But what if you don't have a simple statistical test to do? What if it's complicated? So in that case, I might suggest you unleash your inner snake, as in you should simplify your statistics. That'd be one option. You could simulate your samples, or you could solicit a statistician. So what I mean by simplify your statistics, so again, you might be running something that's more complicated, maybe a two-way ANOVA, but maybe the most essential difference is really just between 2 groups. So you could just look at, a 2-sample t-test style power analysis to get out the most essential difference rather than trying to do a more complicated test. And this could matter for even more complicated designs. So that might be one way. So simplify down to the bare bones. Another thing you could do, this is probably more on the statistician side, is actually simulate your samples. Because sometimes it's hard to like actually tell what you might get out. So it might be good to spell out what you think and actually make some mock data here. So say I expect a twofold decrease in tumor weight between a control and a treatment in mice, and I know a certain standard deviation. I could run some code to simulate data and could actually get mock data that represents that. So I have a control with a mean of 5.28 because there's some noise in that standard deviation, and then a treatment half of that. And I could use that means standard deviation to calculate effect size. And finally, of course, you could solicit a statistician. That's why I'm here for, like, say, TRANSCEND projects. And even if you think I'm not up to snuff here, something that's usually very helpful with statisticians is they might have access to things like paid software like Pass, which I'm trying to get better at. And so if at the very least you can use me to get to Pass. So with that, if you want to dig more, there's certainly a bunch of resources. You can go download GPower. You can, if you're already proficient in R, you could download packages like Power, etc. I have previous trainings on power analysis from more basics that kind of cover the same thing I did here, but with a little bit of a different lens. Or I have more detailed things with GPower, R, or even R with generalized linear mixed models. If you really want, I could pull out, those are multi-video trainings. I could pull out all the links if you want. With that, happy to answer questions, comments, and go into any more details. So thanks so much.
I have a question. Dr. Williamson,
Yeah.
why people say 80% power? Can you elaborate?
Yeah, so that's just again kind of a reasonable threshold because like more power is always better, so I think the resource I have there is like for clinical trials, it might be 0.95. But the more power you have, the more samples you're going to need. And so it's sort of like that balance of like, well, if you had a power of 0.999999, it'd be even stronger, but then you need so much more samples. And it's kind of like a pragmatic cutoff of you might yeah, if that makes sense. If you could get more power, great. But oftentimes, you're balancing like you only have so much time, effort, money, so that might be as good as you can do. If you're lower than that, you might need to rethink your design because it might be underpowered. But that's a good, fairly pragmatic threshold that you can usually meet. So that's why.
Thank you.
Okay. Looks like Dr. Combs, you have your hand raised. Go ahead.
Yes, thank you. So when you are determining the in your example, when you're determining a coin's d-value, you
Yeah.
had the d equals d equals 2 is considered huge. What would you consider what was the definition of large and huge? I
Yeah,
wasn't really
yeah,
sure.
yeah. So those metrics of small, medium, and large, I think, comes from Cohen. Like he was a statistician who kind of developed a lot of these tests about power analysis, sample size. I think in some of his original work, that's what he sort of rules of thumb for like small, medium, and large. And so, it differs by test, but for Cohen's d for a t-test, two sample t-test, it's generally considered zero point 0.2 is small, zero point 0.5 is medium, zero point 0.8 is large, and then I added other terms, like, well, if you're larger than large, you're probably huge. So that's kind of where that comes from. Again, because effect size is the measure of the what how big the difference is in means between your groups, and also factoring how messy or how noisy the data is. I don't know. Hopefully that answered your question. Some of it's just sort of kind of develop rule of thumb, but it does track like real kind of the real relative differences in, say, your means that you're trying to
Okay.
detect.
No, I was just wondering in your graphs,
Yeah,
to me the difference between the two groups was
yeah,
rather small. It was sort of partially overlapping with the standard deviation also
yeah.
overlapping. So I wasn't wondering
Hmm.
why that would be considered huge.
Yeah, well, yeah, because given effect size, you're looking at mean of one versus a mean of two, and so that's a twofold difference, and the standard deviation was, I think, zero point 0.5. So that was just the clarification. Yeah, and so like probably in the real world, like some of those examples I showed where I got like a D of like 5.3 or something for my radon tests, and those were like by eye, like clearly distinguished, like the control and the treatment. And so that, you know, was bigger, and some of that, yeah, goes into even like statistical tests. Something can be statistically significant, but like not very biologically significant or valid, and vice versa. So it kind of depends on really the data and the system you're working on. Yeah, in some places where even though you run the power analysis, you run statistical tests, it says it's significant, but maybe it doesn't actually matter in the real world because that kind of difference isn't really meaningful.
Thank you.
Okay, Dr. Schwartz.
Yeah, Mark, I just wanted to say, first of all, that was great. I loved it. And if anyone doubts the truth of the maxim that the surest way to educate is to entertain, awesome, awesome. I just wanted to just fill in a little bit on Kumi's very important question. I think that part of the answer about how big is big, it depends on what field you're in. And I say this because some of my colleagues who are studying gene induction, if they don't see a threefold induction in a gene, it really doesn't, it doesn't melt their butter. On the other hand, if you are conducting a clinical trial, the general rule is that if you see a 20% difference, a 0.2% difference between a treated group and the untreated or the usual care difference, that is meaningful. So really, the size of the difference depends upon who you are and what your field is.
Can I ask one more question if there isn't anybody else
Yeah, sure, go
asking
ahead.
question? So also the in your repeated measure ANOVA example,
Yeah,
the Rick Astley one,
yeah,
um, so you had calculated the means for each group, each age group
yeah.
to calculate the F value, right?
Yeah.
So what's how did you calculate the means? It's because you had the different time points. Is it all together, or is it at each time point?
Yeah, yeah. So, yeah, repeated measures and over can be a bit tricky. From what my best understanding is, if I was just looking at the differences between groups, so within factors, I think I just need the overall means across between groups. Now, if time is really important for me, um, again, you might be looking at a different question, like, well, is there differences across time or differences across time and groups? Then I know it gets more complicated, and you might have to have means, you know, across, I think, each time in each treatment group. I just kind of took the simpler option, just for showing off. But yeah, it depends on what your research question you actually care about.
Okay.
Because sometimes like a repeated measures ANOVA might be like you just care about difference between groups, but you can't get, as many samples, but you could get less samples and sample them more times. And so that's one way of maybe working with a limitation of smaller sample size, but sort of getting more out of each sample.
Okay. Thank you.
Are there any more questions for Dr. Williamson? Well, that was great. Thanks, Mark. And thank everyone for attending today. And just a reminder to take a few moments to complete that survey. We'd appreciate it. And everybody have a great day. We'll see you again.